Hello students, welcome to Vedantu NEET English. I am Ms. Gopika, your own biology master teacher. I hope I am audible and visible to all of you. Please let me know in the chats if I am audible and visible, and then we can start our class. Okay? Yes, please let me know if I am audible and visible. Quickly put a lot of fire emojis in the chat so I know that we are live. Yes, we are live. Everything is perfect. Perfect. Okay. Awesome. Yes, quickly share the link with your friends and ask them to join. I know there are a lot of students who are lazy to join, but we need to join this class because this class or molecular basis of inheritance is the chapter with the highest weightage. Even 36 marks to 40 marks can come from this chapter. Okay, so there's no way that all of you are going to skip this chapter by any cost. Clear? Okay. Let me just share the link here also, and also quickly smash a like button and mark your attendance so I know that all of you are active here. Awesome. Yes. Okay, good morning. So students, we are going to do molecular basis part 4. I know that is like, ma'am, so many parts. Yes, this chapter is a kind of chapter that can take a 7 hours marathon. Even teachers have taken up to 10 hours, 11 hours in the offline as well. So this is that kind of a chapter. Even in our batches, we take a lot of classes for this chapter because this chapter has to be taught in a more simplified manner, yet, covering all the lines of NCERT. So it is going to be, a kind of a chapter that will take a lot of time for you to understand, but on a long run, if you have watched all this lecture up to the 3 parts and now you are in the 4th part also, you are going to understand everything smoothly. And I will do multiple revisions of this chapter as well because this chapter is very important. Okay, yes, good morning all of you. Okay, those who are new to the channel, welcome you all. And students, look here, today's topic is going to be genetic code, translation and lac operon. So according to our time permits, we'll be doing all these 3 topics. Otherwise, tomorrow at 8 p.m. one more class I've kept where we'll be completing the chapter. HGP and DNA fingerprinting is the plan. Okay, so yes, let's start. First is genetic code. So students, before I go to genetic code, we already understood what is our transcription, right? Transcription means where our DNA strand, right? Double-stranded DNA. This double-stranded DNA formed a single-stranded, okay, single-stranded mRNA, single-stranded mRNA. So this is what was happened so far. What happened was transcription, right? So we have DNA, which is double-stranded, and here we got mRNA. mRNA went through RNA processing because it had introns and exons, right? So here, before we get this mRNA, we got an mRNA that had, okay, that had both introns as well as exons, right? Introns are the ones which will not help. It is the non-coding part. So we had to remove that with a process called as RNA splicing. Okay, so that entire thing we call it as RNA processing. So students, after this, what happened? See, it was simple so far, right? That it was simple that from DNA we made RNA, and it was easy. Why? Because these things were all complementary to each other, right? Let's say in DNA we have A, G, A, T, T, C. Okay, we know that in RNA we are going to get U. A will code with thymine, so that will be uracil. Okay, G will code with C, again U, A, C. So that means what? These people were complementary, and there was something called as base pairing happening. All the nitrogenous bases, we knew the story, what was happening. But when it came from mRNA, like when we have to do from mRNA, we had to do protein synthesis. Okay, when we had to do protein synthesis, do any of you see any similarity with mRNA and amino acid or protein synthesis language? Yes or no? Okay, yes or no? Human health I have not started. Human health and diseases I will start later. Okay, so after that notes you will get. Clear? After that you will get the notes. Clear? Yes, a very important chapter. So listen here. Students, from mRNA to protein synthesis, that means here you are talking about mRNA language, and here you are talking about amino acid language. Okay, AA means amino acid. So amino acid language we will be talking. So here you notice that there is no connection at all. There cannot be any connection, because here I cannot work according to complementary, because here what am I going to get? I am going to get polypeptide chains, right? I am going to get polypeptide chains. So here people had a doubt, okay? Everyone was thinking that, wait, what would be the answer here? What could be the, thing that would connect these people? But definitely these people have a strong connection. Okay, definitely these people have a strong connection. That is when they realize that, wait, whatever codes are there in mRNA, that means whatever nitrogenous bases that you're seeing in mRNA, they are read as codes, and that codes are going to help us in protein synthesis. I hope the 2-3 lines of NCERT is clear to you. So NCERT says that DNA to RNA was a very simple process. Why? Because there was nitrogenous base, there was complementary base pairing, and it was an easy process. But when did the process become difficult to understand? When mRNA and the amino acid language or protein synthesis language did not have any connection. Okay, did not have any connection, and that is where we had a problem that, oh wait, how do we actually convert? How do we actually do translation? So that is why here, students, remember, here transcription was easy. Okay, because it was DNA to mRNA. Here, this was a little complicated. Why? Because one was a RNA and one was a protein, and they both did not have the same language. Is that clear to all of you? Yes or no? Clear? Yes, clear. Okay. Awesome. Shivarajarani, why are you spamming, dear? I understood. Notes will be given after the class. Okay, done. Now, look here. Amino acid language. This is the introduction of how genetic code came into existence. Now, if I say the mRNA, okay, let's just take that mRNA. Okay, we will just draw one straight line. Okay, this is the mRNA. Now, this mRNA, let's say it has AUG, U, okay. Here, let's say it has UAC, okay, and here it has UAA. So this was the mRNA, okay. This is the mRNA. We did not know much about it. We just know that this is the mRNA. Later, we got to know that these three people are going to be read as triplets, and we can call these people as a codon. So this becomes codon number 1, this becomes codon number 2, this becomes codon number 3, and this becomes the 4th codon. Okay. Now, the thing with this is, I have highlighted it in red. What could be the reason? Because these people are not going to code for any amino acid. This becomes, okay, this becomes a stop codon. It becomes what, students? It is a stop codon or a nonsense codon. Nonsense codon. That means they are not going to code for any amino acid, but they are going to help us with termination. They are going to help us with terminating the entire process. Termination. Understood? Understood? Yes or no? Clear? Awesome. Okay. Now, AUG. Now, AUG has to code for some amino acid, right? But why have I written AUG in the starting? Because here, understand, AUG will act as an initiator codon. Initiator codon means it is going to help us with initiation, okay? So, it is going to be initiator codon, or we can say start codon, because we told stop. So, that means we should have a start point also, start codon. But this fellow will have dual function. This fellow will have dual function. Why? Because this fellow is going to code for methionine. It's going to code for the amino acid methionine. Here we have, it is going to code for phenylalanine. Like this. Okay. So these people are going to code for amino acid and help us in the formation of the entire protein synthesis is going to happen, where we have individual amino acids. Now, listen here. This is the introduction of how genetic code conversation came into existence. But how did we come to a conclusion that AUG will code for methionine, or let's say the triplets itself will code for it? That we have to understand by a person who told, okay, I'll just erase this. Okay, now students, this conversation about genetic code, we understood what went wrong, what exactly happened. But here, see, very simple. We have 4 nitrogenous base. What are the 4 nitrogenous base? A we have, G we have, U we have, and C we have. These are the 4 nitrogenous base that is present in our mRNA. Yes, ma'am, these are the 4 that are present in our mRNA. Now, since these 4 are present in our mRNA, but the problem was that we got to know that the total number of amino acids, okay, total amino acids are around 20. So they told us, if you're making a codon, you make sure that we will be able to code for 20 amino acids. So that was a confusion. That is where a physicist called Gamow, okay, Gamow, he was like, wait, there should be a way to decode this. So what did he try? What did he try? One second. He was like, we should be able to crack this code. How we will crack the code? So he cracked the code. First, students, he was like, okay, since we have 4 nitrogenous base, why don't we utilize, okay, why don't we utilize each of them? Let's say this fellow codes for 1 amino acid. I'm just writing 1AA, okay. This fellow codes for the 2nd amino acid. This fellow codes for the 3rd amino acid. And this person codes for the 4th amino acid. Now you tell me what went wrong here? What was the issue here? Tell me quickly, what was the issue? Quick. The issue here was that this was 4 to the power 1. That means 4 amino acids we could code for, okay? 4 codes will code for just 4 amino acids. Leaving how much back? 16 of them are going to be pending. 16 of them were going to be pending. Is that okay? It's not okay, because I told you that you have to code for 20 of them. You have to code for 20 of them, right? You have to code for 20 of them. Are you all understanding? Yes? Tell me quickly in the chats if you're all understanding. Yes, okay. Students, quickly mark your attendance. What is this? Day by day, you people are going down in marking attendance. Quickly smash the like button. Okay. So here we understood that 4:1 is not working. That means, let's say I'm telling A, you please code for 1 amino acid, G, you code for 1 amino acid, you code for 1 amino acid, C, you code for 1 amino acid. It is not working out. Okay. Then we were like, okay, let's try another option where we do 4 to the power 2. 4 to the power 2 means 4 into 4. Okay, 4 to the power 2 means 4 into 4. That means 4 into 4 codons. That means we are going to be able to code for 16 amino acid. Now, what is exactly 4 into 4? You remember that, let's say AU, okay, UG, CU, like this we get AU, UG, CU, like this. This combination we get where we combine 2 nitrogenous bases, okay, 2 nitrogenous bases, and we form a code. That time we realized that here, even if I do this, okay, even if I do this, I am getting 4 pending. Yes or no, students? I am getting 4 pending. Is that okay? It's not okay, right? So I have to make sure that I'm going to get all the 20 done. Okay, all the 20 done. Okay, yesterday's notes is not updated. It will be updated in some time. Okay, I've already shared the notes with them. It will be in the description box. So 4 pending will be there. That is when George Gamow was like, wait, I kind of understood how to decode this. That is 4 to the power 3 would work. That is 4 into 4 into 4, which is going to give 64 codons. 64 codons, and they will be easily able to code for 20 amino acids. But again, 64 codons when we have 20 amino acids. That is where we got to know that the codons have an ability called as degeneracy. That means one codon can code for many codons will code for 1 amino acid. That means serine will be coded by 6-7 amino codons. So that is where this ability of codon came. Okay. 1 amino acid will be coded will be coded by multiple codons. Okay, multiple codons. So this capability came. Because this capability came, students, this 64 codons became easy because 61 codons, okay, 61 codons were coding for an amino acid, okay, they were coding for an amino acid, okay, and the three of them, okay, the rest three of them, they became the stop codons. They became what? The stop codons, or else called as the nonsense codon. That means they are not going to help us with coding for amino acid, but they are going to help us with termination. Okay, they are going to help us with termination. Is this clear to all of you? Please let me know in the chats if this is clear. Okay, so he was the one who told that your codons have to be triplet. Okay, codons are triplet, and it has to be triplet. Okay, it has to be triplet, then only the entire thing is going to work, where each of the codon is going to code for an amino acid. Awesome. Yes. Hi Jerusha. Okay, this is the basic understanding of your codon. I hope this is clear. Now students, we have to study about some additional scientists. Okay, that also we will study here. So genetic code, we understood that every strand, let's say this is an mRNA. Okay, this is an mRNA. Okay, can all of you give me any 2-3 amino acid codes quickly in the chats? Any 2-3 amino acid codes can you give me? Anything. Okay, whatever you feel is correct, you just give me. Yes, smash the like button quickly, guys. Yes, okay. So here we go. We start again with AUG. So I'll put AUG in green because it is the start codon. Okay, green line says us that, okay, it is time to start. Okay, AUG. Okay, then we will just continue. We have U, 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 A. Okay, and then maybe we will put G, A, C. Okay, you just tell me which are the codons. Okay, UAG, UAG, UGA. Okay, those are stop codons. I want codons which have the amino acids in them. Come on, give me the amino acid ones. Quickly in the chats. It's easier. Just open that box and just give me which are the ones who are going to code for what. You just don't give me just the amino acids, okay? Because I have not by-hearted the entire box. I have not by-hearted the entire box. Okay, now here we extend a little more. Yeah, and here we write the deadline. The deadline would be UAA. Okay, something like this. So look here, students, I have written one code here, and let's start writing what is going to happen. So all these places, please remember, in all these places there is going to be a tRNA that will be attached. Okay, we usually draw tRNA like this. Okay, there will be a tRNA which will be attached. Now this tRNA will attach and code for what? Here they are going to code for methionine, right? Here they are going to code for methionine, and also it is a start codon. Here what are they going to code for? Here they are going to code for phenyl. Okay, phenylalanine. So this is how your amino acid formation is going to happen. Okay, and here let's say they are going to code for leucine. Okay, then here what are they going to code for? They are going to code for valine. Okay, valine they will code for, and here obviously termination is going to happen. That means these people are going to stop. Okay, so this is how the entire codon is going to help, students. That is, this 3 will start. Okay, so remember here also, in translation also, we have to go through the process of initiation. Okay, we have to go through the process of initiation. We have to go through the process of elongation, and here we have to go through the process of termination. Clear? I hope all of you understood how genetic code is going to work. Okay, how genetic code is going to work. Okay, now look here. Hargovind Khurana synthesized homopolymers. So what he was doing, let's say he was working in a lab and then he understood that we can easily form, okay, we can easily form something like this, okay, U, 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 U. That means these are homopolymers, right? So look here, same there. This is one, this is one, this is one. So that ended up saying, okay, we can form homopolymers. Okay. At the same time, he also worked and we could understand that we can also form copolymers. Okay. How will you form copolymers? Look here, all of you. Let's say U, G, U, G, U, G, U, G, U, G. Now this becomes copolymers. Yes or no? Copolymers. U, G, U, G, U, G. This becomes copolymer. So he formed homopolymers and we could also form copolymers. That means we have 1, 2, 1, 2, 1, 2, 1, 2 like that. Okay. Now, Marshall Nirenberg developed a cell-free system for protein synthesis. That means he was like, no need all this cell and everything, all this complication is not required. In a test tube itself, I will make sure that I can form a protein and give. So that is how these people finally found a system where we do not have to worry about central dogma because everything can be done in the lab. Okay, we will learn a little more detail about them here. Look here. Okay, so Hargovind Khurana developed a chemical method to make artificial RNA with known base sequence, like replicating the same base or combining specific base. Okay, this helped in studying which codon makes which amino acid. That's why I told you he did this combination. He did just one combination, okay, specific combination that led to the formation of homopolymers. He also utilized another technique where he combined like this, okay, and he got copolymers. Done? Okay, now Marshall Nirenberg created a cell-free system, a lab setup using test tube where the protein synthesis, okay, where the protein synthesis could happen outside a living cell. Can you imagine? Protein synthesis, I have been telling from that time that students, this is important, this is happening in our body. Just imagine, no cell at all, it is happening in the lab. Okay, it is happening in the lab. This allowed the scientists to test which RNA sequence produced which amino acid. So this allowed the scientists to understand that which RNA sequence would produce which amino acid. Now, Saverio Oho discovered an enzyme called polynucleotide. Okay, polynucleotide phosphorylase. This was asked one of the year's question, which that could bring RNA molecule without a DNA template. Okay, that means you take a test tube. Okay, you take a test tube. Okay, you have all the codons. Let's say U, A, G. Some codons you have here. Okay, U. Some codons you have. This phosphorylase. Okay, this phosphorylase will be present, and here we understand that there will be no DNA. Without a DNA, we will be able to form. So that could build an RNA molecule without a DNA template. This helped the scientists, okay? So without DNA, you could build a RNA. Just remember their names, questions can come. Okay, this helps scientists create RNA with known sequence for experiments. So these three people are important for you to remember. Hargovind Khurana, Marshall Nirenberg and Saverio Oho. Enzyme also you have to remember. That is your phosphorylase. Now look here students, this is how the genetic code came into existence. How? Look here, you have U. That means normally in your Punnett square you study with two boxes, right? First position and second position. Here you can see first, second, third position. That's how we will be able to get a triplet. Simple, no? How will you get a trio? By having three friends in your gang, right? So here you can see U, understood? UUC, UUA, okay, UUG. This is how you get it. Okay, next one, how will you do? UCU. So C will come in the between. UCU, UCC, UCA, UAG. So if you look here, serine is coded by almost 4 codons just in one box. Like that, again, serine is coded here multiple times. So that means what? One amino acid is coded by multiple codons. Okay, that is where the nature of degeneracy come into existence. Is this clear so far, students? Please let me know in the chats. Is this clear so far? Yes, no, anything to be repeated? Quickly. Why can't I see your chats? I think here it's very slow. Chats are very, very slow. Done? Why is attendance not increasing? Huh? Why is the attendance not increasing? Okay, yes. Okay. Students, call your friends, yeah. Such a nice class and the attendance is so less. Okay. UUC, done. So this is how we are going to get codons. Here also you can see leucine is coded here. Okay, this many times. Then histone, arginine, okay, lysine. See how many repeated times they are being coded. So this shows that the codons are degenerate. Now here comes a very, very important question that can come for all of you. The salient features of genetic code. Okay, the codon is triplet. Students, what is it? The codon is triplet. We already understood, ma'am. Codons are going to be this way, UAA. Out of that, 61 codons are going to code for amino acid and 3 codons are going to act as a stop codon. That means they're not going to work or they're not going to code for any amino acids. Okay. Now, some amino acids are coded by more than one codon, hence the code is degenerate. This question has come multiple times, so please remember it, that more than one codon will be coding for same amino acid. Okay. Now, the codon is read in the mRNA in a contiguous fashion. That means what? You're not going to read it like, let's say, UAC, UUU, AUG, which is not supposed to come, so AGU, okay, UUC. Now look here, students, this is a codon. Now how are you going to read it? You're not going to read it as UAC, UAC, comma, UUU, comma, UAGU, comma, UUC, full stop. No, you're going to read it in a continuous fashion, because remember, when the tRNA is reading also, let's say even if one codon is not there, it is still going to read it continuously. It is not going to be like, oh wait, UAC should have been there. It will not. Okay, it will just continue the reading machine. Okay, now the code is nearly universal. That means from bacteria to human, you will notice that some of the amino acids are coded by the same set of codons. For example, from bacteria to human, UUU would code for phenylalanine. Some exceptions to this rule is the mitochondrial codons in some protozoans. AUG has dual function. What is AUG's dual function? Mainly it will act as a start codon, but it also will code for methionine. So it will code for an amino acid, at the same time it also acts as a start codon. Now, UAA, UAG and UGA are stop terminators. Okay, UAA, UGA and UAG and UGA. Okay, so UAA. So how can you remember this? So I usually remember that A is coming in 2 positions in the center and G is coming in 1 position. Okay, so you can remember like that, or you can also remember you go away. Okay, you go away, you away go, and you okay, away. So you can remember something like that. It's not a trick, but it is something that I've heard. Okay, so that is clear. Genetic code is clear, perfectly clear. All of you tell me. So we understood how the codons became triplet. We understood how we can utilize the nitrogenous base and how these codons are going to help in the formation of a protein synthesis. So that was the missing link between your mRNA and your protein synthesis, which was finally understood when genetic code came into existence. Okay, now here comes, students, there's 2 other concepts that is very important for you. One is called as frame shift mutation. Okay, frame shift mutation and deletion. Okay, or like deletion. So frame shift mutation happens when you do an add-on of any of the nitrogenous base. So here they have given an example with a normal example they have given, but remember, if it was in a codon position, how it would be. So here, students, Ram has red cap. Any problem with the reading shift? No. The reading shift is, reading shift is perfect. Reading shift. Like how we say frame, like that, the reading frame is perfect, right? I'll write here reading frame. Okay, reading frame is perfect. That Ram has red cap. Anyone has a problem in reading that? No. But let's say the moment I decide to add this B here, I decided to do what? I added one nitrogenous base, B. Okay, So this led to what? This led to addition. Now what is this? Addition, or I can say insertion. Addition of or insertion of one NB. That means one nitrogenous base. Now what will happen? Do you think tRNA will stop and tRNA will be thinking, wait a minute, from where did this B come? No, tRNA will not do that, because tRNA knows codons are triplet. It is continuously going to read 333. So here what is happening, students? Definitely the codon is going to read RAM has, so it is going to be the first codon, second codon, BRE, third codon, DCE, fourth codon, and whatever is continuing, it is going to code. Now the problem here is frame shift mutation has happened. That means what? The reading frame has got affected, right? The reading frame has affected, which will lead to mutation. Now here, students, what is happening? Here you remember, here we have added one more letter, okay? We have added one more letter. So again, insertion of two NBs, okay? Two nitrogenous bases. Now when I inserted two nitrogenous base also, my reading frame is changed. RAM has BIR, EDC and AP. Does it make sense? No ma'am, it does not make sense at all, because I cannot read it, right? So here also insertion of two nitrogenous base has almost changed the entire reading frame. Now let's see here, we have inserted the third letter. So insertion of, now what has happened? Insertion of 3NB. So when I insert the third nitrogenous base, I can read it properly as RAM has big red cap. Does it make sense to all of you? Yes or no? Quickly tell me in the chats. Does the statement make sense to all of you? Come on, quick. Does the statement make sense to all of you? Quickly in the chat box. And those who haven't marked the attendance, quickly do that also. Faster. Possible? Make sense? Yes ma'am, it makes sense. Ram has big red cap. That means what? Before he had a red cap, now he has a big red cap. Makes sense. So here, what does it mean, students? This gives us the conclusion. Okay, this gives us the conclusion that we cannot do addition or insertion of nitrogenous base in the improper numbers. It has to be a triplet. Okay, so addition or insertion should happen in multiples of 3. Okay, in multiples of 3. Why? So that we can avoid frame shift mutation. So this addition, okay, this addition or insertion has led to frame shift mutation. Clear to all of you? Easy? Okay, it has led to what? Frame shift mutation. So whenever you are adding a nitrogenous base, you have to make sure that you are adding it in multiples of 3. So why? Because we are going to read the codon in triplet form, and it is not going to be read in, however, let's say tRNA will not stop and think that, oh wait, I should have read it this way. No, it is going to continuously read it. You will understand how when I teach you translation topic. Okay. Now, students, same thing can happen if I delete it. Okay, let's say RAM has EDC AP. It was given. Okay. Now I am going to delete the letter. Okay, I am going to delete E. When I deleted E, what happened? You just look at the reading. Okay, again, RAM has DCAP and whatever it continues. Here EDCAP. So now next what I am going to do, I'll be removing D from it. Okay, I will delete D. So first I deleted E, second I deleted E. Now you see RAM has cap. Okay, RAM has cap. That means what? Here when you delete also, it is understood that you need to delete in the multiples of 3. Okay, so deletion mutation can happen. Okay, because of deleting a nitrogenous base. Deleting a nitrogenous base can cause deletion mutation. That means mutation that is led because of deletion of a nitrogenous base. Okay, deletion also can lead to mutation. Again, if deletion has to happen properly, it has to be all the 3 codons that has been removed. Okay, clear to all of you? Is this clear? Quickly give me a thumbs up in the chat. Yes, 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 yes. I can see few S's. Few S I can see. Done? Okay, okay. Okay, students. Now here itself, you can just study about sickle cell anemia. So in a normal DNA, we would have GAG. Okay, GAG would be there. Let's say GAG would be there. That GAG would code for what? Here we would have had C, T and C, right? This would be the code. C would be the code, right? This is normal DNA. Now a person who has been affected by sickle cell anemia, okay, the person who has been affected by sickle cell anemia, what is going to happen? Sickle cell anemia, what is going to happen? Students, this should have ultimately coded for glutamate, okay? The amino acid should have been GLU, glutamate. But here what is happening in sickle cell anemia, here we are getting GUG and that will code for C, U will code for A and C. Okay, so this is what we are getting. Okay, we are getting something like this. So here what is happening? GUG will code for what? GUG will code for valine. So that is how sickle cell anemia happens. So by this you should understand that even a small change, okay, even a small change in one nitrogenous base can lead to mutation, okay, can lead to mutation and it can lead to such disorders. I hope, yes, glue replaced by valine. Good, Kirti. Okay, yes, valine. Okay, GUG. Done. This is your deletion and your mutation. Now coming to translation topic. Okay, translation. So students, before I directly go into translation, you have to understand during this entire process, I have already taught you this about the different types of RNA, RMT 1, 2, 3. There also I told you that when we talk about this RNA does not, see, it might not be the genetic material for all of the organism, but it does do other jobs, right? Like we can see here, we have a tRNA. What is tRNA going to do? This tRNA is going to act as an adapter molecule. Adapter molecule. Now, what is this adapter molecule? See, students, when we got the mRNA, okay, when we got the mRNA, we knew that we have to convert this mRNA to protein. Again, we understood the language, we understood the genetic code, but did you see anywhere that mRNA can attach protein or attach amino acid? Can it attach amino acid? No, ma'am, it cannot attach amino acid. Let's say protein itself, they will be able to attach themselves. No, they also can't. So we understood that between them there is a problem or there is some gap that has to be bridged, and that is where we brought tRNA into place, who is going to act as a adapter molecule in helping in, okay, who is going to help us in the entire translation process. Entire translation process. That means they are going to help us add the amino acid and help us in the formation of the polypeptide chain. So now, talking about tRNA, if you look at the structure of tRNA, if it was a straight strand, it would look different, but it is complementary, so it has come together and it looks like a double strand, okay? Here you can see, students, in tRNA, there's mainly, for you to remember, there is one two regions that you have to remember. The region that you're seeing down, okay, the loop that you're seeing down is called as the anticodon loop. So this is how it will look in 2D and this is how it will look in 3D, okay. Anticodon loop is present here, and on the top, this region, you will notice that this is an amino acid acceptor site. Amino acid acceptor site. So down we have an anticodon loop, top we have an amino acid acceptor site. Now what does this mean? Let's just draw it here, okay, let's just draw it here. We have one loop here, okay, we have one extension here, we have one more loop here, and here we will have the main loop, which is our anticodon loop. Now this is how, here remember, in every loop there will be almost 7 unpaired bonds, okay, 7 unpaired will be there. Now here we will have a short arm, this is the short arm, okay. Here we will have your DHU loop, here we will have T5C loop, okay, yes, and this is our anticodon loop, anticodon loop. Ma'am, what is the use of this anticodon loop, ma'am? How are these people going to work? Very simple, students. Down, what am I going to have? I am going to have an mRNA, right? I am going to have a mRNA here. Now, let's say on the mRNA, I have A, I have U, and I have G. Now, I know AUG is there, but this tRNA has to add the amino acid. So, anticodon is going to form. Okay, so this is the codon. It is going to form anti or complementary structures here. That means A will code with U, U will code with A, and G will code with C. It will form UAC, automatically letting your amino acid acceptor site to understand that, okay, we need to start the process and we need to add methionine. So understood how this process is going to happen? Let's say from down one person is reading and from top the other person is attaching. It happens in every company. Let's say a company that is producing something, okay. Down the person will be reading the label, what and all you should add into a box. Let's say we are making a science kit for the kids, okay. So he's telling conical flask, he'll add the conical flask. Okay, he's telling beaker, he'll add the beaker. Glass rod, he'll add the glass rod. And the box will get packed and it will move. Same way, down, let's say AUG is there. Anticodon loop will be like, wait, it is UAC. Immediately amino acid acceptor site will be like, okay, I need to start the process and I need to add methionine. Next codon, UUU, automatically it will be replaced as what? AAA. Automatically here it will be like, okay, phenyl. So this will entirely form the polypeptide chain. Is this clear to all of you? Please let me know in the chats. Clear? So students, in 2D, it will look like a clover leaf model. Okay, in 2D, it will look like a clover leaf model. And in 3D, it will look like an inverted L. Excuse me. Yes, done? Understood? Okay, so sorry. Yeah. So here, this is what is about your tRNA. Now look here, a few lines of NCERT. From the very beginning of the proposition of the code, it was clear to Francis Crick that there has to be a mechanism to read the code and also link it to the amino acid. So when we understood about genetic code, we understood that, wait, there should be someone who can read it, and according to the reading, we will be able to attach amino acid, because mRNA is not capable, protein cannot do the job by itself, so we need a middleman. Okay, now because amino acid has no structural okay, amino acids have no structural so in some words, some days, no, it does not just come in my mouth. Okay, okay, no specialities, okay, no structural specialities, and read the code uniquely. He postulated the presence of an adapter molecule that would okay, that would one hand read the code and on the other hand would bind the amino acid. Yes or no? Let's just imagine tRNA like this. One hand it is reading the code, one hand it is attaching the amino acid. So doesn't it do the same job? Yes ma'am, it does the same job. One hand it is reading, one hand it is attaching the code. Okay. Now to specific amino acid, the tRNA then called snRNA. Why? Because it was a soluble RNA, was known before the genetic code was postulated. However, its role as an adapter molecule was assigned much later. So first people used to call it sRNA or soluble RNA. Later, once this genetic code, everything came into existence, they understood, wait, this soluble RNA has much more job of becoming like an adapter, and that is how tRNA became an adapter molecule famously. Done? Okay, tRNA became an adapter molecule. Now students, question will come in such a way, they will ask you the 5' end, 3' end, they will ask you where is the amino acid attaching. Okay, you should tell amino acid is attaching on the 3' end. Okay, where are they attaching? The answer will be it is attaching on the 3' end. Okay, so this kind of questions will come, remember. Now look here, this was the codon AGU. What did we get in the anticodon? A will code with U, G will code with C, U will code with A. Okay, here what we got? We got UAC. So UAC, what will be the opposite? U will code with A will code with U, C will code with G. Correct. So these are forming the anticodon. So on one hand they are reading the codon. Okay, on another hand, they are adding the amino acids. Is this diagram clear? Now we will, I know it's very bright. Okay, I don't know if I can reduce the brightness. Now tRNA has an anticodon loop. Okay, it has an anticodon loop that has bases complementary to the code, and it also has an amino acid acceptor end. Okay, to which it binds to the amino acid. tRNAs are specific for each amino acid. For initiation, there is another specific tRNA that is referred to as initiator tRNA. So remember students, tRNA is going to be specific. It is not going to be like, oh, I don't have a tRNA, can we share? No. Every amino acid, every codon will have a specific tRNA. Why? Because it will be bringing its amino acid. Like it will be coming with its amino acid, no, once it reads the code. So it will be specific. So like that we have initiator tRNA. Okay, now here there is no tRNAs for stop codons. In figure 5.12, the secondary structure of tRNA has been depicted that looks like a clover leaf model. That's what I told you. 2D looks like a clover leaf model. 3D looks like a inverted L. Look here, the structure. So it looks like an inverted L. Yes or no? See, like this. Okay, inverted L structure 3D will look like, and 2D looks like the clover leaf model. Now students, if you type lucky, okay, if you type lucky in your phone keyboard, there is a leaf that comes, which is like four portions are there for that leaf. That leaf is called as the clover leaf. Okay, clover leaf, we also call it as a lucky leaf. Clear? Lucky leaf. So here in actual structure, the tRNA is compact molecule which looks like an inverted L. Any doubts so far? Because we will be starting translation, and only if you understand tRNA structure, translation is going to be easy job. Okay, easy job. Just put lucky in the chat box. We'll see if that leaf is going to come. I will try, okay, first. See this one. It comes. Can you see? Lucky. Yes, very good. That is the clover leaf model because it has 1 loop, 2 loop, 3 loop, and on the top amino acid. So it will look like a clover leaf model. Understood? Now, translation. What is exactly translation? So, translation, students, here, remember, we had our mRNA. Okay, we got our mature mRNA after all the processing. Now, this mRNA has to become protein. Okay, it has to form protein. Ma'am, but why, ma'am, protein is so important? Students, it's very simple. Protein is so important. Why? Because even including factors like your eye color is determined by your proteins. So, protein plays an essential role not just in, genetic inheritance, but also it plays a very role in all our body activities, right? So, that is why protein synthesis is so important for us. So, from mRNA to our protein synthesis. So, here we have our RNA and here we have protein synthesis. So, here we have a protein chain. Now this process will be called as your translation. Translation. Now, while translation happening, you remember RNA processing, everything has happened. But here comes a very important thing, students. Normally, we have studied that everything is happening inside the nucleus. Yes or no? Anyone has a doubt in that? Yes, ma'am, everything was happening inside the nucleus. That is why we used to say that, okay, inside the nucleus, this is happening. Now, inside the nucleus, what was happening? Inside the nucleus, we got, okay, I should have drawn a bigger nucleus. One second. Okay, this is the nucleus. So inside the nucleus, we had our double-stranded DNA. Okay, we had our double-stranded DNA. From the DNA, we got a single-stranded mRNA. Yes, this much was correct, right? mRNA. Now, students, this mRNA, I will have to send it to the cytoplasm. Why? Because if my translation will happen in the cytoplasm, my other two processes, including replication, everything was happening in the nucleus. But now I need to transport this mRNA to the cytoplasm. Clear to all of you? Yes or no? So here we already have done capping and tailing. So you can remember here we have our methyl. So I will just write it. One second. Methyl guanosine triphosphate on the 5' end, and here we have poly-A tail. Okay, poly-A tail, which is on the 3' end, right? We have already done that. So now we have to transport our mRNA from the nucleus to the cytoplasm for translation to happen. So if they ask you the location, you should be in a position to tell the location, right? Yes, Jerusha, all Conjus people have joined, so they will not do anything. Okay, so here protein chain formation. Now students, one amino acid to another amino acid, there should be a peptide form. Okay, peptide formation happening. So let's say this is one amino acid. Okay, we'll take same methionine and phenyl only. Wait. Okay, we have these two proteins. Okay, why is this not working? 1 second, students. Okay, these are two proteins. Okay, inside this we have, let's say, methionine, MET, and here we have phenylalanine. Now I am planning to form a polypeptide chain, right? So that means between these people, okay, between these people, they have to form a polypeptide chain. Okay, I'm drawing a heart. They have to form a peptide bond. They have to form what? A peptide bond. But how will this peptide bond formation happen? For this peptide bond formation to happen, I really need some charge, some energy I need. So translation, the first step for translation is the amino charging of your tRNA or amino acylation of your tRNA. Okay, so this is clear. I've just drawn a heart there, so you understand that peptide bond have to happen. And here also, if I continue, the next peptide bond formation should happen. Clear? Now this diagram we will come back, students. First we will try to understand translation in detail, and then we will come back to this diagram. Okay, all of you are refreshed. No one is tired? No one is tired or something? I hope all of you have the energy. We will be only doing translation. Lacopron I'll do tomorrow evening. Okay, because I feel most of the students have gone to school and college. Okay, so that's why. Done? Okay. So the first step in translation, okay, students, here remember in translation also initiation, elongation, termination, everything happens. But here in translation, the first step that happens is activation. Okay, activation. We can also say amino acylation, but we can say activation better. So we can write activation of amino acid. You'll be thinking, what, ma'am, what is activation of amino acids? See, students, we have this entire process is going to happen with the help of energy. Okay, so here activation of amino acid means what? I need to make sure that the amino acids are having the energy to go forward, and they have to go and combine with tRNA. So for that, I am going to do amino acid plus ATP. Okay, this is energy, right? So I'm adding energy here. And see, this process is actually more in depth. It is there, amino acid synthesis. So we'll have an enzyme as well. So here I'm going to get amino acyl. Now students, remember here, when I'm forming this, I am basically having one hope, that these people are going to continue and start the charging of the tRNA. Clear so far? Yes or no? All. You want to know the enzyme name? I can write the enzyme name. Otherwise, we will not write the enzyme name. Okay. So it's a complex formation here. Why AMP? Because we utilize this energy. Because here, one phosphate, it will come out, right? So that phosphate, we will write it here. Plus PP. So here we have amino acyl. Here we use an enzyme. Okay. Amino acyl synthetase you can use. Enzyme. Okay. So if you want to remember, you remember the enzyme. Otherwise, no force. You don't have to remember the enzyme. Okay. So the enzyme is going to be amino acyl tRNA synthetase. I'll tell you why it is so important for us to charge our tRNA as well. So look here, synthetase. Okay, something is wrong. Okay, now students, look here. After this, we are going to take this fellow. Okay, who is that? Amino acyl AMP complex. Okay, we're going to take that. And the second process is going to be, charging of tRNA as mentioned in your NCERT. All these notes will be available on Vedantu NEET English Telegram. At the same time, it will be also available in the description box. The link will be there. So make sure that you download the link. Okay. So next is going to be charging of tRNA, students. What are we going to do? We are going to take your amino acyl AMP complex, and we are going to now introduce a tRNA. Okay, we are going to introduce the tRNA. So this way we are going to do charging of tRNA. What are we going to do? Charging of tRNA. Why tRNA should be charged? I'll tell you. Okay, I'll tell you. So students, here see, it is simple. Okay, amino acyl tRNA complex will be formed. Okay, if you can remember, well and good. Otherwise also, this is just to make you understand better. Okay, amino acyl tRNA complex you will get. Complex. So this way your tRNA has now got charged. Okay, plus AMP. AMP will be thinking that, oh no, now I am become a headache, so I will just leave. Okay, I will just leave, and that person will leave, and this will lead to charging of our tRNA. So let's say we draw tRNA like this. Okay. So now the tRNA has got charged. Now why is it so important for charging the tRNA? Okay, let's just draw the waves like this. Okay, students, why is it so important for us to charge the tRNA? Because see, if this tRNA is charged, okay, for charged I am drawing this tRNA is charged. What is the job of tRNA? It has to add the amino acid and it has to help in the formation of the polypeptide chain. Now let's say this tRNA is charged and here we have another tRNA. Okay, we have another tRNA. Now this, let's say this tRNA also is charged. Okay, this tRNA also is charged. It will automatically allow these two tRNAs, to form a peptide bond. To form what? A peptide bond. If I form this peptide bond only, I am going to get an entire polypeptide chain, right? So we need two charged tRNA to come close. When they come close, automatically they will form the peptide bond. Is this clear? Is this clear, students? Can you at least tell yes or no in the chats? What is the kind of, things that you people will not even tell yes? Tell me. So, amino-acyl tRNA complex will be there. So, when two charged tRNAs come together, automatically peptide bond formation will happen. Now, this is the main two processes that happens in translation. Activation of amino acid and charging of tRNA. You can also say amino acylation, okay, because you know amino acylation is happening. So, you can say amino acylation of tRNA. Okay, amino acylation of tRNA. One step if you write also it's fine, but this is the step. Okay, so here we have got charging of tRNA. We understood what is translation now. Now, how exactly this process is going to happen? See students, we already know that we have a tRNA to help, which has an anticodon loop. Okay. Okay, here a short arm, and then here we have a 5' and a 3'. And here the amino acid formation is going to happen. With the tRNA, okay, we already have a tRNA. And with the tRNA students, we are going to take the help of ribosome. What are we going to take the help of? A ribosome. So look here, we are going to take a help of a ribosome which has a larger subunit and which has a smaller subunit. Okay, one larger subunit and one smaller subunit. So here, okay, this is the larger one. Okay, this is the larger one. This is the smaller one. So in prokaryotes, it will be 70S. So it will be 50S and 30S. It is not the totaling, it is the Sweatberg's unit. So you cannot consider sediment unit. You will not be like, oh ma'am, 50 plus 30 is 80. No ma'am, how did you put 70? No, it is set. It is a different way of calculating them. Okay, it's actually the sediment factor, or we say Sweatberg's unit. So now students, the smaller and the larger subunit of the ribosome should come together for the entire process to happen. That takes me to exactly how translation happens. I am teaching every step very, very, simple and this way. So I hope all of you, later also when you're watching it recorded also, you please let me know if you understood all of this. So look here, let's say we have our larger subunit, okay? And students, a question has been asked that larger subunit or smaller subunit, which one helps in the translation to start? You will write the answer is smaller subunit, okay? Now look here, this is what we have. This is our larger subunit and we will have tRNA. Now on this larger subunit, you will see there are 3 sites, E site, P site and A site. This is important. Many times what happens, your NCERT does not explain this and you do not understand how these people are moving. So can you see 3 sites? E site, P site and your A site, okay? Now what is exactly happening? Here students, your E site is like an exit site. That means where the, okay, one second. Okay, now the colors are there so you can see the sites properly. So look here students, E site is the exit site. Okay, where the tRNA will exit, okay, after giving the amino acid. On the other side, we have the P site, which is the peptidyl site. Peptidyl site, okay, and we have the A site, which is an acceptor site. Okay, amino acid acceptor site. So we have one E site, P site and A site. Now what is going to happen? Okay, one second. Okay, now students, here, sorry. Now look here, what is exactly going to happen here? Let's just erase these people. Okay, now I want all of you to remember one thing. Okay, so here we have EPA site, right? EPA site. So let's say one tRNA is waiting, okay, one tRNA. So I'm drawing tRNA like this, okay, like with one hook, the anticodon loop. And let's say one tRNA is having a UUC, okay, UUC. That means what? It is trying to read the mRNA. So how is the reading going to happen? Your tRNA, so keep aside everything, reading everything you keep aside. Now you just imagine A site is going to accept, okay. P site is where the peptide formation is going to happen. So P site is the place where the peptide formation is going to happen. What happened? One second, students. Okay. Okay. P site is where the peptide formation, that means the entire polypeptide formation is going to happen. So here, first let's say, I can't see the light. Okay. Yeah. So first P site, let's say one person we are going to attach. Okay. Let's say any amino acid you are going to attach. So here we will have our mRNA. Okay. We will have our mRNA. Okay, I'm just removing the smaller this one, so no confusion is there. Now students, let's say we have here AUG. Okay, we have AUG. Now on the P side, we will have what? We'll have the tRNA. Okay, we will have the tRNA having what? Ma'am, we will have U, A and C. Correct? UAC. That means what? We have to add the methyl, right? So here we will add the methionine. That T will be bringing all of this. Okay, that T will be bringing all of this. So here we go. This board, no, some issue is there. Okay. One second, students, some issue with the board. Okay. So here we have a tRNA. Okay, tRNA here. It will have what? A will code with U. Okay. U will code with A. G will code with C. So here at the other end of the tRNA, what will we have? We will have the methionine already attached. Right? We will have the methionine already attached. So now this is clear to you. So now after attaching this methionine, what will they do, students? It will be ready to leave. Okay, it will be ready to leave. And then the next person will be waiting. Okay, next person will be waiting. Let's say here we have U. So here we will see on the acceptor side, you will see another tRNA. Okay, I'm just erasing the site so that you know. Okay, see here on the acceptor side, we will have another tRNA who is waiting. Okay, who is waiting? What is he waiting with? He is waiting with A. He's waiting with what? A. Why? On this side, he will have what? He will have phenyl. Okay, he will have phenylalanine. So, the moment this fellow exit, okay, the moment this fellow exit, the next fellow will come and sit and the chain is going to continue like this. Okay, we'll have meth, okay, methionine. We will have phenyl. Okay, so like this, the chain is going to continue. Is this clear to all of you? Is this clear to all of you? Tell me. Yes, Kirti, I'll do that very soon. Okay. Is this clear? Come on. Okay, now I will just draw it here down. Okay, so what is going to happen after this? Okay, so here we have our mRNA. Okay, we have AUG, we have UUU. Okay, now here students, what is going to happen? Here after attaching in the P position, okay, in the P position, you are going to see what? You are going to see one chain of methionine. Okay, you're going to see one chain of, red means methionine, okay, one chain of methionine. Then you're going to see one chain of phenylalanine, right? Correct? Yes or no? Now next person will be waiting. Okay, next person will be waiting in the A site. And remember, the ones who already came, okay, the ones who already came will exit from this site. The tRNA will be exiting from this site and one person will be waiting to join here. So here we will have, let's say GUU, okay, GUU. So here who will be waiting, students? One tRNA will be waiting. Okay, I'm just going to write it like that only. So let's say one tRNA will be waiting with what? It will be waiting with G will code with C. Okay, U will code with A and U will code with A. So it will be waiting to come in here and then move here. And if it does that, the next you will be able to see valine here. So here he will be carrying Okay, here another tRNA will be waiting like this, and he'll be having G will code with C U A A. Okay, he'll be waiting like this, right? He'll be waiting Y to come here, and from here he'll move here, and on the other side he'll be having valine to attach. Valine to attach. Done? All of you, please tell me if this is clear. Okay, this is clear. So like this, students, the entire polypeptide chain formation will happen. Okay, polypeptide chain formation will happen. So this is how the general site is. So one will be waiting to accept, one will be, peptidyl will be already attaching the amino acid, and exit, they will be exiting. So this is how it happens. One person comes, attaches, continues, goes forward. Okay, so this is what we have to learn here. Done? Done? Little tricky, but I hope this diagram helped. I will just erase this diagram because I feel this diagram might confuse all of you. Okay, let's see if we can do something about it. Okay, we can. Okay, so here I will draw it a little more neat so it's clear. Okay, so first we have exit site, then we have peptidyl site, and then we have acceptor site. Okay, and down we will have the mRNA. Okay, so we start with AUG, and then we have UUU. Okay, UUU. So here what is happening? First we have to attach the methionine. So here methionine will be attached. How methionine is going to attach? Because here you will have U, here you will have A, and here you will have C. Okay, which will be brought about by your tRNA. Okay, which will be brought about by your tRNA, and it is going to help in connection with your amino acid. Done. Now the moment this person is done, the other person will be already waiting. The person will be waiting with what, students? It will be waiting with A, and A. So he will shift here. So this guy will shift here, and this attachment will continue. Okay, this attachment will continue. So here we will have phenyl. Okay, phe. Got it? This is how your entire polypeptide chain formation will happen. Clear? Easy? Okay, so we will get, ultimately we will get a polypeptide chain that will have, okay, that will have methionine, okay, that will have phenylalanine, okay, that will have valine, and like this the chain will continue. Now students, this chain formation, remember here one tRNA is there, right? So here, let's say here we have tRNA. Okay, this is the tRNA I am drawing because full loop I can't draw. So let's say your tRNA, this is the anticodon loop. First tRNA was what? UAC. Okay, this tRNA is already charged tRNA, and it has a methionine here. Now this tRNA came and it was in the peptidyl site, right? Now the next tRNA, okay, the next tRNA comes. Next tRNA comes, and what will it do? It will be waiting here, right, to move. So that time it will have what? A, and A. So when these two are there, this is also charged, right? This is also charged tRNA. Automatically, students, the peptide bond formation will happen. Peptide bond formation will happen. Clear? So this is how the whole process happens, and that is how we get the entire polypeptide chain. Otherwise, how for peptide bond formation, we'll have to come and call another person and come. So here we automatically get the peptide formation. So this is how it happens. So remember, this fellow will become the initiator tRNA, and remember tRNAs are specific. Okay, initiator tRNA. Now you will have a question, ma'am, how does this entire process stop? Right, you would have thought, no, how does this entire process exactly stop? So students, here the entire process is going to stop when on your mRNA, okay, this blue color is your mRNA, no? On your mRNA, we will have the stop codons, okay, because stop codons do not have do not have any tRNAs to code. So automatically this will lead to, okay, this will, wait, I'll move this here. Okay, so this will lead to stopping, because these people are going to be the stop codons. With the stop codon, students, we also have something called as untranslated regions. What do we have? Untranslated regions, which are UTRs. Now, where are UTRs present will come as a question. So let's assume this is your mRNA, and on this we will have, let's say here we have AUG. Okay, AUG we have, and here we have UAA. Okay, stop codon we have. So this is our 5'N, this is our 3'N. So here, okay, here we will have our UTRs. That means before the start codon, on the 5'N, and after the stop codon, okay. Okay, after the stop codon, clear? So this way we will be able to easily reach termination. So termination is taken care by your stop codon. With that, we also have untranslated region. Untranslated region means those are a small region which will not go for translation, but they will increase the efficiency of translation because they are present right before the start codon and right after the stop codon, okay, on 5'N and on 3'N. Can you remember this much? Yes, possible? Can you remember? So this is your ribosome, okay? Done? Okay, board got stuck. Let's wait for a minute. I hope it will work, students. Let's just wait. Okay. Oh, wait. Something happened. Okay, is it better now? Okay, did you guys get stuck by any chance? Okay, this is done. Okay, and this is also understood. Yes, understood the entire formation. I hope there's no confusion. Okay, after this automatically this is happening. Okay, and there I have told you about the peptide formation, and this is how termination happens, students. So here remember, your UTRs are actually, full form is what? Untranslated regions. Okay, untranslated region. What are they going to do? They are going to increase the efficiency. Okay, increase the efficiency of translation, but they will not take part in translation. Okay, in the translation, and where they will be present, already you know, they'll be present on the 5'N and in the 3'N. Done? Yes, clear? So the process is clear now, how the P side and then they move to the E side. So remember, one person will be waiting here. As this person attaches, this person leaves, this fellow will join, next person will be waiting, and here you will have your mRNA. Okay, here you will have your mRNA in between. Like this, your mRNA will be there, where you will have the 5'N here and 3'N here. So this is the entire structure of your, this is entirely, we can call this as the translation unit, because this is the smaller subunit, this is the larger subunit of the ribosome. And students, only if the smaller subunit comes, then only the entire translation will start. Okay, so this is the ribosome plus the tRNA and plus the mRNA. Clear? So this is how the entire translation happens. Now going back to this diagram, look at this diagram. All of you just look here carefully. Okay, look here carefully. You will notice that, see, this is the P side. Okay, this is the P side, the peptidyl side. You can see the GCA coding is happening, tRNA is attached, and this entire chain was formed by multiple other tRNAs who came, went, left, came. So here you can see this is the exit side. Okay, this is the exit side. This is the exit site, because you are here, you can see U coded with A, C coded with G, C coded with G. So this becomes the exit site. Now because once it already added the alanine here, it left, okay, it exited. So here exit happened. Now here you can see this is waiting again, okay, this is waiting. This fellow has already formed, so he's going to transfer the entire chain to this tRNA, and this tRNA will come here, this fellow will come here, understood? So here let's say 1, 2, 3. So 1 is going to exit, okay, 2 is going to come to 1's position, and 3 is going to come to 2's position, understood? So that is how the entire formation of the polypeptide chain happened. So remember that when these two tRNAs are together, they have already formed the peptide bond. That is why this exchange is happening. Otherwise this exchange won't happen. Now this transformation, this movement is happening because the peptide bond formation has already happened. Clear to all of you? Yes or no? This is what is happening. This is the diagram, okay? So I hope diagram is clear. Now we'll just go through NCERT. Okay. Translation refers to the process of polymerization of amino acid to form a polypeptide. So what is it? Process of polymerization of amino acid to form a polypeptide. Okay. Now the order and the sequence of amino acids are defined by the sequence of bases in the mRNA. So the order and sequence of amino acid depends on what are the bases that are present in the mRNA. This word though we understood, right? Now the amino acids are joined by a bond which is known as the peptide bond. Okay, it is known as the peptide bond. Formation of a peptide bond requires energy. Okay, to form the peptide bond, what do you need? Energy you need. Therefore, in the first phase itself, amino acids are activated. That is why I told you activation of amino acid is going to happen. Activated in the presence of ATP. The reaction, I hope all of you remember. This is the reaction. Activation happened when amino acid was combined with ATP. It formed aminoacyl AMP complex and PP. That is the energy that came out with the help of an enzyme called aminoacyl tRNA synthetase. Okay. Synthetase, some spelling mistake is there, no students? I'm not able to understand. Today my spellings are not, words are not. Okay, now okay, and linked to their cognate tRNA, a process commonly called as charging of tRNA or aminoacylation of tRNA. So first activation of amino acid happened. That activation will lead to aminoacylation of tRNA, so or charging of tRNA. Okay, so that combination is very important for us to remember. To be more specific, if two charged tRNA, see this is what I told you, if two such charged tRNAs are brought close enough, the formation of the peptide bond between them will happen, and it is more favored. Okay, favored energetically. The presence of catalyst would enhance the rate of peptide bond formation. The cellular factory responsible for synthesizing protein is going to be ribosome. We already know ribosome is the cellular factory that helps in protein synthesis. The ribosome consists of, that is why from nucleus we are transporting it into the cytoplasm. Okay. Now look here. The ribosome consists of structural RNAs and about, student, this question has come in one of the years. Ribosome consists of structural RNAs. Other than that, they have about 80 different proteins. Okay, this is important, and it has come in the previous year NEET. Okay. Okay. Now, in its inactive state, okay, in its inactive state, it exists as two subunits. That means a larger subunit. What are they talking about? The ribosome, okay, and a smaller subunit. When the small subunit encounters an mRNA, the process of translation of mRNA to protein, okay, mRNA to protein begins. Therefore, two sites in the large subunit for subsequence. So, here, this is the question, okay, this is the question. When who comes and attaches, does the process happen? When the small subunit encounters an mRNA, the process of translation of protein begins. So, here, question again, important. NEET PYQ. Okay, NEET PYQ. Who starts the or who initiates the reaction? Clear? Yes or no? Come on, students, yes or no? Quickly. Okay? Quick, 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 quick. Hungry? Okay, done. Now, there are two sites in large subunit for subsequent amino acids to bind and thus be close enough to each other for the formation of subunits amino acid to bind and thus be close enough formation of the peptide bond. Now, the ribosome also act as a catalyst. Okay, the ribosome also act as a catalyst. 23S RNA in bacteria is an enzyme ribozyme and for the formation of the peptide bond. Now, a translation unit in mRNA is the sequence. Okay, look here. A translation unit in mRNA is the sequence of RNA that is flanked by start codon AUG. So, to start the process, we need AUG, and to stop the process, we have stop codons, okay, and codes for the polypeptide chain. And mRNA also have some additional sequence, okay, that are not translated, but they are referred to as untranslated region or called as UTR. Okay, untranslated region or called as UTR. Now, the UTRs are present at both the ends. So, students, this is again a question. See, the UTRs are present at both the ends, that is 5' end before the start codon and 3' end after the stop codons. Why? To increase the to increase the efficiency of translation process. For initiation, the ribosome binds to mRNA at the start codon AUG that is recognized only by the initiator tRNA. So here, look here, for initiation to continue, okay, for initiation to continue, what will happen? You all need break, very small topic I have, almost done. Okay, now look here. For initiation, the ribosome binds to mRNA at the start codon AUG that is recognized only by initiator tRNA. That means the start codon will be only recognized by initiator tRNA. The ribosome proceeds to the elongation phase of the protein synthesis. So this is what I told you. Your translation also, okay, translation process also happens in 3 steps. Initiation, where you will have an initiation tRNA, then we will have elongation where we are forming the polypeptide chain, and then we will have termination where you will have your stop codon, okay, where you will have the stop codon. So this process all happens, okay, this process all happens. Now, during the stage, complexes composed of an amino acid linked to the tRNA sequentially bind to the appropriate codon in the mRNA by forming complementary bases with tRNA anticodon. The ribosome moves from codon to codon along the path, okay, along the path. So that is very, very important. So here, where is it? Okay, just duplicating this, okay. Oh, some problem with the board. Okay, students, here I'm just going to add about the translation process, okay. So here, remember, translation happens in 3 steps, okay, 3 steps. What is the steps? First one is called as initiation. First one is called as initiation. So here, students, I'm going to write who and all are responsible, okay. Initiation, you will have start codon, okay, you will have a start codon that is going to be your AUG, okay, and with the start codon there is going to be an initiator tRNA, okay, initiator tRNA, Okay, initiator tRNA will be there. Then we will have our second step, that is going to be elongation. Elongation means what? You're forming the polypeptide chain. Okay, elongation means what? You're forming the entire polypeptide chain. Okay, so here you will have who? You will have the entire formation of the polypeptide chain. Okay, and here students, the peptide bond formation, charging, everything would have happened, and then the peptide formation would have happened. Okay, then the last process is going to be termination. Okay, termination. So in termination, what is going to happen? Obviously the stop codons are going to be present. Okay, stop codons are going to be present. That is your UAA, you go away. Okay, you go away and you away go. Okay, so UAG. Now with the stop codon, students, you will also have something called as release factor. Okay, again these are all there, so the termination can happen. You will have something called as the release factor. If you check your NCERT, you will find it. And you will also have UTRs, which are present in the 3' end. Okay, you will see them, which are present in the 3' end. Clear? So this is important. Okay, what happens in all the three processes. I hope this is clear. Yes? Awesome. So I will be stopping the class right here. Okay, I'll do Lacopron. I have the notes, but then many of the students are missing, so I will just do it later. So tomorrow 8 o'clock I will do HGP fingerprinting, Lacopron and DNA fingerprinting together. So yeah, see you all tomorrow's class. Now students, before going, most of you are asking me about ma'am, how is the pro batch? Okay, what is happening in the batches? So here you have students, so just go here. Here you will be able to see link. Okay, if you see here, you will get flat discount on NEET 2026 and 27 batches. So if you are taking a 2 years course, okay, just so imagine you're taking a 2 years course for NEET. That is, you're in 11th standard, so 11th and 12th you're going to write, you're going to prepare for NEET. Then you have a 20,000 flat off on the pro batches. Let's say you're taking a 1 year plan. That means you have already completed 11th, you're right now in your 12th, you want 1 year course, then you will be able to take this. Now you'll be asking, ma'am, what's the big difference between prime and pro? Students, prime is a affordable batch, but see, content, everything we deliver at the best, even on YouTube, even in the affordable batches. But in pro, what happens, you will have lot of individual attention, just like how you feel in offline. You will have competition, that means many other students also will be there. You will have meet with everyone, that means all your cameras will be switched on, all of you will be unmuted. It will be like a web class where I am teaching you. But here what is the difference is, you will have proctor mode. Proctor mode means, all of you remember during the COVID time we have all written exams, right? During that the schools used to have a proctor mode. That means if you look away from the screen, the proctor would give you a warning that you are looking away from the screen. If after multiple warnings also you're not coming back, or you're not focused on the class, the call would directly go to your parents. Same thing here also, the direct call will come to you, and that would lead to your parents also being aware that okay, online does not mean distractions. Here we have a proper system in place where we will make sure that you are not distracted. So if you're a student who genuinely has only one thought, NEET MBBS, then you can definitely go for this. So first you can book a demo, students. You can just see how the entire batch thing is going to happen. So just enter your name, enter your grade. Okay, you could be a class 12 student. Which board are you right now studying in? What's your target need? What type of course are you looking? Long-term batch, online long-term batch. Select the medium of language. This many mediums are happening. Okay, so English medium you put, and then you put your name and phone number and proceed. This way you will be able to get a one-on-one counseling session where they'll explain you what and all is there in the thing, and you can also ask your questions, because you're paying a huge amount. You would want to know the details of it. Okay, so that's it for today's class. I hope all of you enjoyed the class. I am little disappointed with the crowd, but it's okay. Tomorrow evening I'll be taking at 8 o'clock. So those students who have missed this class and who are watching it recorded, join me tomorrow for Lacopron, HGP and DNA fingerprinting. So see you all students. Till then, take care. Bye-bye.